for those of you who were here a couple of weeks ago, this is using the same strategy of using a Google Collaboratory notebook in order to analyze some of this data. So it should look pretty familiar. So if you could just let me know when you're able to get that up on your computer, if you're interested in following along. Yeah? Okay, cool. So if you remember, you can either run these blocks of code by pressing the play button or by hitting Shift Enter. But before you even do that, you need to, oh, I thought that I put this, you need to copy this. Oh, this is actually the wrong notebook. I'm sorry, 1 second. Yeah, it does. Yeah, so it was weird. It wouldn't show me any of the figures, and I like looked on Stack. You need to recopy your notebook into like a new file and whatever. So I was like, okay, whatever. Oh my God, I can't type anymore. Why? I'm sorry. I know that I had this pulled up somewhere else, but okay, here we go. Okay, so this should be the one. It still says defunct. Oh my God. Wow. It wasn't showing output figures, and no matter how many times I restarted the kernel, it wouldn't work. Okay, so I will relink this real quick. Yeah, because you had to copy it over, but the one that I started editing, this one has more edits on it. So yeah, here you go. I will delete that old one. Okay, I'm sorry about that. Okay, so yeah, first step, copy this notebook, which it seems like you guys have already figured out how to do. And we'll reinstall Scanpy. We had it installed last time we met. Oh my God, this is driving me crazy. Why is it blinking? Are you done? Okay, so reinstall Scanpy. You can press the play button or hit Shift Enter. And then we have data already accessible for you, but you need to follow these instructions in order to make it accessible. If you did this last time, you don't need to do this stuff again, but you will need to mount the data. So run this block here. It'll prompt you to log in and enter an authorization code. Please use your UCI email address for that, because I think we extended privileges, viewing privileges to all of those people. Okay, let me know if you have any problems with that. Unfortunately, we have to go through this like startup every time because it doesn't save any of your progress. Okay, in the interest of time, I'm going to be able to stay afterwards, but I know that some of you probably have things to go to. So I'm going to go ahead and run these two blocks. There's information that you can read on here. This is a downsampled data set because we want to look at a lot of like what changing a lot of these parameters does. And in order to do that in a setting like this one, we have to reduce the number of cells that we're looking at. So this is actually one sample from the data set that we're going to be working with all quarter. And yeah, so it's this sample, this AD11 sample. Okay, so we talked a little bit about normalization. So I think that it's pretty standard fare to normalize your data before you perform dimensionality reduction on it. Again, I'm not as much of an expert as PJ is, but usually you see these steps, you normalize. Here we're normalizing to 10,000 counts per cell, and we're also taking the log transformation of each of the counts. So you can go ahead and run that and it'll work really quickly. Yeah, so yeah, that's one of the difficulties of trying to teach the single cell analysis is how much it like loops back on itself. So we're trying to give you a logical flow of it while realizing that there is actually no logical flow. Yeah, so the next step is finding highly variable genes, which Paige talked about. So here we have, let's see. And there's actually, I meant to put another part in here that shows you what's in the data structure, but we can put that in for the next time we meet. So we have 28,000 genes in our data set. And so the first step that we can really use in order to reduce our dimensionality is by computing and subsetting on which genes are highly variable. And you can do those two things by running these lines of code here. And there's a bunch of information about how it works up here that you don't need to read right now. And then we can go see that, oops. We can go see now that there are 3,000 highly variable genes, which we discussed that variable information is sometimes the most important information to preserve. So this is why we find highly variable genes here. There's some figures you can make. I'm going to skip over that. But let's get to actual principal component analysis. So another method of kind of like data normalization / cleaning that you can do before you finding principal components in your data is scaling it. So this is scaling it to unit variance, which I'm not going to explain what that means. But this is all it takes in order to find the first 50 principal components in your data set. So this is now taking the dimensionality of your data from 3,000 highly variable genes down to 50 linear combinations of your gene expression space that are represented by these first 50 PCs. And a nice way to kind of guess how many principal components you might want to use in your data is actually by looking at the variance ratio for each of your principal components. Here, these are kind of overlapping one another, but essentially what you're looking at here is how much information is contained within each principal component. So you can kind of see that there's this like knee shape here, which if you're familiar with doing like a UMI per barcode cutoff sort of thing, you're looking for a knee here. You kind of want to minimize the number of PCs that you use because that'll be easier computations while maximizing the information that you get. So maybe for this experiment here, we could pick like PC 20, which I know that you can't read that, but that's what this one says here. So this is kind of an informative plot to help you figure out what hyperparameters to use. So yeah, there's some information that explains that here. And so what do the first two principal components look like if we plot them and maybe plot the expression of a couple of marker genes on them? Well, you can see that the marker genes are like localizing to specific parts of PC space. So it is indeed a useful form of dimensionality reduction. But if you were to try to put this in a paper and say like, these are my clusters or whatever, they'd be like, what are you talking about? Like it's like an octopus with 3 legs. So not super useful for like kind of our intuitive way of visualizing the data. And so that's where our friends nonlinear low-dimensional embedding come into play, such as UMAP or TSNE. Here I'm only going to go over UMAP because TSNE is so much slower, as Paige mentioned. There's some, this particular article here that's linked is actually the same one that Paige linked. We found the same one, which is pretty useful at explaining the theory behind it, although you don't need to worry about it too much. And also, as Paige mentioned, the UMAP embedding relies on finding the nearest neighbors or computing the neighborhood graph in your data set. And so this is another thing that is like a tunable parameter, is how many neighbors to consider. And there's information about what that does in this text here, but again, in the interest of time, I will kind of gloss over it. And you'll also notice here that another parameter is the number of principal components you'll use. So I know I said 20, but for some reason here I decided 30. But again, it's all tunable. So if we go ahead and run this, it'll take a couple seconds. Is anybody actually running this stuff on their computer and able to get it to work? Yeah? No? Are you having trouble? No, not your UCI. You need to click the link and then log in as your UCI account. Yeah, sorry. And yeah, so for people with like 4 Gmail accounts like me, I have to be specific about which one I pick. Okay, so yeah, actually, if you wanna go around and help, that'd be useful. So here's our UMAP. Now we're getting something that looks a little bit more clustery than this here. And so now the next step is to actually cluster this data. And so we're going to here use the Leiden clustering algorithm. And you don't need to know that much about clustering, but one of the parameters that I really want to kind of, I want to emphasize is the resolution parameter. So the cluster resolution parameter can be used to basically increase your granularity when defining clusters or decrease it. So a higher number means you will get more clusters. So if you're working with a sample where like cell subtypes or even different substates of the same cell type matter a lot, you'll want to use a higher cluster resolution. And if you're working on a data set where you only want to like broadly define like a few different cell types, or if you're working with a data set with a really homogeneous cell population, you might want to use a lower resolution. But in practice, of course, since analyzing single cell data seems to be so cyclical, this can entail you clustering it one way, going to look at marker genes, and then realizing that you may be missing cell populations that you want, or you may be trying separating out cell populations that you want to aggregate. And so the way that you can address this is by adjusting the resolution parameter. So here we're going to cluster with resolution 1. So you'll see now we're able to color this UMAP with different sets of clusters. And that's where this portion of like the kind of explanation portion of this notebook ends. If you guys have any questions on this, I have some more stuff to show below, but are there any questions on what I've shown already? Were you able to get it to work? Yay! Okay, great. Any other people trying to get it to work that can't get it to work? Okay. Okay, so so that you can understand what the impact of changing some of these parameters when you're doing the analysis, I've made a couple of functions below that will loop through a set of parameters that you give it and display what the actual effect of it is on the UMAP and the clusters. So if you run, we'll have to reload the data because it's going to reprocess it each time. So you can reload the data and then make sure to run these 2 blocks too, because that will make these functions accessible to you. And then don't rerun these things because there's already output displayed for it and it takes a while. But essentially, here you can vary the number of highly variable genes that you're considering, the number of PCs that you're considering, so principal components, the number of nearest neighbors you're going to use to compute your neighborhood graph, and the resolution that you're going to use when creating your clusters. So here's an example where I'm varying the highly variable genes. So I'm trying 1,000, 2,000, and 3,000, and trying 5, 10, and 30 nearest neighbors. And I don't know if I could tell you what this is doing differently based on this plot, but if you stare at it for longer than I did, you could probably figure that out. I think that the easiest one to really see the difference in is when you vary the resolution. So here we're using a lower resolution, 0.5, and here we're using a higher resolution, and you're seeing that you're getting more clusters in the resolution 1 or in the higher resolution column. I guess if I were to say something about the UMAP for the PCs, it's that this kind of looks like fluffier, almost like less compact and like looking at populations of cells on the UMAP. What was that? Mm-hmm. Yeah, okay. So here's another one, which is varying nearest neighbors and resolution, and another one for some reason. So if you want to go ahead and run this yourself, you can just add another code block here. And then you can pick 2 parameters to vary, because I don't want to deal with making a 3-dimensional figure. So say we want to test like number of PCs and highly variable genes. I don't know if I already did that. We can just change this to PCs and HVGs and then give it like a list. Say we want to use 2,500 here, and maybe we want to try using 30 and 50 here. So then if you run that, it'll take a while, because now it's making, well, it's actually only making 2 plots here, but for the one that was like 3 by 3, that one took a while. Yeah, so this is kind of a good way that you can get an idea of how changing these different hyperparameters will affect your data. And there are other parameters that you can change that I didn't make accessible for everyone here, which is unfortunate. But if you go look at the function documentation in Scanpy, it's really, it's useful, and there are people who have put together like tutorials on like how these things affect your final UMAP and clusters. Yeah, I guess that's pretty much all I have, unless there are any more questions. I'm sorry that this section was rushed, but again, you have the notebook and you can take it home and run your own data on it if you want. Any more questions? Mm-hmm. Yeah. If you're analyzing your data as a first pass, you can absolutely just use the tutorial, but keep in mind what changing some of these things does. So if you're wondering where your specific population of cells is, maybe try changing the cluster parameter. Maybe try increasing the number of highly variable genes that you're looking for, because those might have not been included in the highly variable set before. Yeah, and of course, like you can always reach out to your community who might have suggestions. It's difficult to develop an intuition for this. And really the only way you do is just by doing it a lot. Yep. I don't know if Paige wants to try to take that question, so.
So maybe,
Here.
maybe understanding this actually get more cells in collaborate. Somehow, some people believe it's more joyful. Because there are a few neighbors active, so it's not so. One way to explain that, one way to try to change that, I think the basic is there's no real guarantee. So empirically, we need to try it.
Yeah, that's a good explanation, because the number of nearest neighbors, or the neighborhood graph, will define how your UMAP is constructed. So if you use more of them, more cells that are further apart from each other will be connected, and therefore it will influence the validity of the global structure more. Yeah, so that's something that people say a lot, is like, just don't look at the global structure on the UMAP or the t-SNE, look at the local structure. UMAP does it a little bit better, but in reality, we should not be making like quantitative claims based on how our UMAPs appear. Yeah, it's a nice visualization, but if you're saying like cluster A is far away from cluster B, don't do that. Yeah, otherwise Lior Pactor will come down from the heavens and smite you from the earth. Any other questions? If you're running through this notebook later and you have any problems, feel free to message me on Slack. I will upload these talks as a video on YouTube as well, and make the slides available. So you'll have everything. All right. You guys can leave. Thank you for coming.